Hello, everyone. My name is Gurjat Basra, and I'm the Manager of Patient Programs, Research, and Advocacy at Lymphoma Canada. I'm pleased to welcome you all to day 2 of our national conference and to our first live educational session of the day titled What's Coming Down the Pipeline in Regards to Lymphoma Therapies. We appreciate that so many of you have been able to join us live today. And before I introduce today's speaker, I do want to go through a few housekeeping items as usual. So this session is being recorded and the recording will become available following the conference. Additionally, this Zoom software does not allow you to access video features or turn on your microphone. So at the end of the presentation, we will try to take a few questions should time permit. And with that said, I would like to introduce our speaker. Joining us today is Dr. Shannon Murphy, who is a hematologist and assistant professor at Dalhousie University and practices at the QE2 Health Sciences Center in Halifax, Nova Scotia. Prior to joining the Division of Hematology and Hematologic Oncology in Halifax, Dr. Murphy completed her internal medicine and hematology training at Dalhousie University, followed by a one-year clinical lymphoma fellowship with BC Cancer in Vancouver. Her practice focuses on lymphoma and chronic lymphocytic leukemia. So thank you for joining us today, Dr. Murphy, and you can get started as soon as you're ready.
Great. Thank you very much, Gurjat. So thank you so much to Lymphoma Canada for the opportunity to speak this afternoon. Today, as Gurjat rightly said, we're going to talk about what's coming down the pipeline, so a little bit about emerging therapies for lymphoma. Just some disclosures. I have previously received speaker fees and honoraria, but there was no pharmaceutical support, financial or otherwise, for this presentation. Our objectives for this afternoon will be to review some treatments that are showing promise for lymphoma treatment. So these are not treatments yet that are widely available, but they are up and coming. And we'll also take the opportunity to discuss some new combinations of established treatments. So maybe some treatments you've heard of being used in new and interesting ways. Tiny caveat, there's no way that we can cover it all. There's so much exciting research happening and irons in the fire that we certainly wouldn't be able to cover all of it. So what I've tried to do is to get a selection of emerging therapies across disease sites as much as possible. There's definitely more information available for some therapies than others. So some of the discussion around particular agents might be richer than others, but as it becomes available, we try and share what we have. And just a note to say, keep your ears to the ground, because certainly there are lots of important studies and therapies that are emerging, so more excitement to come. So let's start with the new and exciting. And I'm going to start with something that you may have already heard about. So we'll talk a little bit about Bruton's tyrosine kinase degraders. So a Bruton tyrosine kinase degrader, or a BTK degrader, is a new class of drug that targets and eliminates the Bruton tyrosine kinase protein. And BTK is a key enzyme that's involved in B cell survival. So BTK helps B cells grow and survive. Overactive BTK allows lymphoma cells to keep growing, and turning off BTK can slow or even stop cancer growth. BTK inhibitors, drugs you might already be familiar with, like acalabrutinib, zanubrutinib, ibrutinib, they act like an off switch for BTK. Degraders instead tag BTK for destruction. So BTK degraders actually recruit the cell's waste disposal system, which is the ubiquitin proteasome system, to destroy the BTK protein. And this is an important mechanism of action because it can help degraders to be effective even when there are resistance mutations present, because instead of blocking communication with BTK, it's physically removing the protein. So very interesting. So you'll hear a lot about BTK degraders, and you may have heard it in the context of chronic lymphocytic leukemia or small lymphocytic lymphoma. And because they can be effective in the presence of BTK resistance mutations, which can be seen particularly in the setting of continuous BTK inhibitor use, which is a common treatment for CLL. But these drugs will likely also have a role in treating other B-cell malignancies, and they're also studied in that space. So I'm going to focus on their role in lymphoma treatment for the purposes of our talk today. And the drug in front of you, BGB 16673, is a BTK degrader being investigated in relapsed and refractory B-cell lymphomas, as well as in CLL and SLL. Also to the right, you can see a lovely pictorial of the degrader and how it engages the cell's waste disposal system to send BTK for destruction here through this ubiquitin and proteasome pathway. So there is an ongoing phase 1/2 study called Cadence 101, which is a clinical trial looking at the efficacy of BJB 16673 in mature B-cell malignancies. The study so far is showing promising responses in relapsed and refractory Waldenstrom's macroglobulinemia, marginal zone lymphoma, and follicular lymphoma. And I have a couple of snippets. So as we get information from various conference presentations. So in ASH of 2024, we heard that among patients, so we had 27 response evaluable patients with Waldenstrom's treated at that point with BJB 16673, and they were showing an objective response rate of 81%. And they had a major response rate, which is a partial response or better, of 74.1%, and a very good partial response rate of 25.9%, which is a very nice early response. We then got an updated ICML early in 2025, back in the spring, that showed as of March of 2025, in response evaluable patients, the overall response rate in marginal zone lymphoma was 50%, still small numbers, so 50% of 20 patients, so 10 patients, but responding. And it was not quite 42% in patients with follicular lymphoma. And of those patients with marginal zone and follicular lymphoma, 2 with marginal zone achieved a complete remission and 1 complete remission in follicular lymphoma. So some early data showing that there can be efficacy there, which is encouraging. And I will say just a quick word about the safety profile, and I'll try to do this with each of the agents that we're talking about using the information we have available. But it's very important when we're considering emerging therapies. Of course, we want to know how well the drug works. We want to see that it's effective, but also really important to know how well people can expect to feel while taking it. So among the Waldenstrom's patients, there were certainly adverse events experienced. So we'll describe them as treatment emergent as adverse events. So we have adverse events that cropped up while on treatment. And most patients will experience some adverse event. So people who experienced any adverse event, it was 93% of patients. But not a lot of high grade, so grade 3 or higher adverse events. So about 40.7% of patients had higher grade, and about 26% of patients had serious treatment event adverse reactions. The most common adverse event with this drug was neutropenia, and often we'll see in hematologic cancers, cytopenias, so low white blood cells, low platelets, low red blood cells can be quite common, and neutropenia or low neutrophils increases the infection risk, so that's an important side effect. In follicular and marginal zone lymphomas, common adverse events also included fatigue and contusion, which is just a fancy way of saying bruising. Upper respiratory tract infections were also pretty common, especially among the follicular lymphoma patients. So of course we have to keep in mind at this point numbers are quite low, but still an important signal. Researchers didn't report any incidents of atrial fibrillation, hypertension, or major hemorrhage, and those are important because when we think about drugs that interact with BTK, so acalabrutinib, zanubrutinib, ibrutinib, those types of side effects are things that we're seeing. So researchers are particularly vigilant looking for those. Also, thus far, there were no events of febrile neutropenia, so fever and neutropenia, which can be, of course, an emergency. We send patients to the emergency department with febrile neutropenia. And there were no treatment emergent adverse events, so nobody had a toxicity that caused them to need to reduce their dose of drug or to stop taking the drug. That's encouraging. However, one patient did die with a treatment emergent adverse effect. So someone experienced septic shock. It was considered related to disease progression, but they were on treatment at the time. And then one other patient died of progression of their disease. So important for us to be aware of that. This is another BTK degrader with some evidence of efficacy in relapsed refractory lymphoma. So this one was called NX-5948, and you can still find information on it using that abbreviation. But it does have a name now, so it's called Bexobrutideg, and it's currently under investigation, but showing some promising results, again, in Waldenström macroglobulinemia. Similar to BJB16673, these new agents, and we talked about it in various disease states, right? Waldenstrom's, marginal zone, follicular lymphoma. They're often looked at in multiple disease sites at once when they have a mechanism of action that's expected to be effective. And so they're often, you'll have a large trial with sub-studies that will then present rolling data in each of the disease sites as it becomes available and informative. So we get little snippets in the different disease sites as various sub-studies of the trial progress. So here, for relapsed refractory Waldenstrom's patients on study with Bexobrutideg, data that was available as of March of 2025 on 19 responsive evaluable patients. So emerging therapies, we're often dealing with low numbers initially, but it did show an overall response rate of 84.2%, including 2 very good partial remissions and 17 partial remissions or major remissions. So good responses, no complete remissions yet, but looks like good activity and still early days, so encouraging there. Looking at the common adverse events, similarly early, but the most common side effect seems to be diarrhea, low platelets or thrombocytopenia, purpura or contusion, so bruising or a purpura is sort of a surface collection of blood without actually leaking blood, and petechiae, which are you may have already seen on yourself or a family member, little tiny pinpoint dots that can result from a pressure in situations of low platelets, sometimes from an increased bleeding risk from a medication, and purpura really is just when all those little tiny dots coalesce together into one big one. There were 2 patients who stopped the drug for adverse events. There was no atrial fibrillation, again, looking for those toxicities that we associate with agents that interact with BTK. And again, very early. So the median duration of treatment so far for these patients was 3.8 months. So certainly lots more information to come, but a good early snapshot. So we're going to change tack a little bit, and we're going to talk about ROR1. So an ROR1 targeting agent is showing some interesting activity in diffuse large B-cell lymphoma. So ROR1 is receptor tyrosine kinase-like orphan receptor 1, and it's an oncofetal protein that's present on many cancers. So oncofetal, it's present physiologically prior to birth, and then it disappears. And pathologically, it can re-present in the setting of some heme cancers and some solid tumors. Zilovertamab vedotin is an antibody drug conjugate, which comprises a monoclonal antibody that recognizes extracellular ROR1, a cleavable linker, and then the antimicrotubule cytotoxin monomethyl auristatin E. And so that monomethyl auristatin E, it's a mouthful, but you might recognize that drug conjugate from such established treatments as brentuximab vedotin and polatuzumab vedotin. So zilovertamab has promising activity against large B-cell lymphoma, and it is being studied in both the upfront and the relapsed refractory settings on the Waveline clinical trials. So in Waveline 003, this is a phase 2-3 study looking at zilovertamab plus rituximab, gemcitabine, and oxaliplatin, which we abbreviate as R-GEMOX, which you may have heard of before. And I realized looking at this slide, I haven't said much about R-GEMOX, but I will dig into it a little bit later. And this is for relapsed and refractory diffuse large B-cell lymphoma. So the phase 2 portion is now complete. And at the recommended phase 2 dose, the overall response rate was 56%. 8 patients achieved a complete remission. There was 1 partial remission, and a median duration of response at that time of 8.7 months. We're talking short follow-up, small number of patients, median overall survival not reached, and a 6-month overall survival rate of 78.8%. So a nice place to start. It's now proceeding to the phase 3 part of that trial, which is going to look at zilovertamab R-GEMOX versus GEMOX. So going head to head with that salvage regimen for relapsed refractory diffuse large B-cell lymphoma. So it'll be very interesting. And then the Waveline 010 trial is looking at upfront treatment of diffuse large B-cell lymphoma. We've had upfront treatments trying to look at ways that we can improve on our standard of care, R-CHOP, for newly diagnosed diffuse large B-cell lymphoma. And we have had a number of studies in the past looking at additions to R-CHOP. Here's one that we're looking at now. So zilovertamab with R-CHP. So it's R-CHOP without the O. Obviously, O stands for vincristine, but they have some overlapping toxicity. And so no O, just ZVRCHP. No data yet. Those early days of recruiting, but will be very interesting to see how that goes. When it was studied in the relapsed refractory setting, so early, but looking at common side effects thus far, most patients, 98% of patients had some adverse event, the most common being diarrhea, nausea, anemia, and low platelets. The higher grade toxicities, about 65% had a higher grade toxicity, and they really primarily were low blood cell counts. So low neutrophils, neutropenia, low platelets, and anemia or low red blood cells. There were 2 patients who had to stop their treatment from side effect, both treatment related. And there was 1 patient who died from sepsis that was treatment related. These were all in the same dosing cohort, and we'll see how things go from here. Okay. ROR1, very interesting. So now we're going to dig in and talk a little bit about epigenetic therapy, which is a really fun topic. And you may have heard about epigenetic therapeutics before. Epigenetic therapy is referring to drugs or treatments that are used to modify gene expression without changing the DNA sequence itself. So that process is reversible. And by targeting epigenetic mechanisms like DNA methylation and histone modifications, these therapies are trying to correct abnormal gene expression patterns that are associated with diseases and particularly with cancers. The idea is to try to reprogram diseased cells to a more normal state. So epigenetic therapy can target things like DNA methyltransferases. So drugs that inhibit DNA methyltransferases can reduce DNA methylation, which can be useful in treating cancers where tumor suppression genes have been silenced or turned down by hypermethylation. So some examples of DNA methyltransferase inhibitors that you might have heard of include azacitidine and decitabine. They're used more often in myeloid cancers, but there is some evidence in certain lymphomas. Targets for epigenetic therapy also include histone deacetylases or HDACs. You may have heard of an HDAC inhibitor. And drugs that inhibit HDACs can increase histone acetylation, which can lead to a more open chromatin structure that allows for gene expression. So examples of HDAC inhibitors that you might have heard of include belinostat and romidepsin, which have some evidence in T-cell lymphomas. And then another target can be histone methyltransferases. So they block enzymes which are involved in gene expression and cell division. They add methyl groups to histone proteins. So it's a type of epigenetic modification that can control gene activity. And we're going to talk a little bit about histone methyltransferase inhibitors with some evidence in lymphoma. So valametostat. Valametostat is a potent dual inhibitor of the enhancer of ZEST homolog 1 and 2 enzymes. And I've forgotten to include the word enhancer here. The E stands for enhancer, so EZH1 and 2 enzymes. So EZH1 and 2 are histone methyltransferases, and they add methyl groups to a particular histone, H3 lysine 27 or H3K27. And that's a mark that silences gene transcription by compressing chromatin. So by inhibiting EZH1 and 2, valametostat prevents this repressive methylation, which can reactivate the expressions of genes that are involved in fighting cancer. So for a good cause. So valametostat, you might have heard of before. It's not a brand new drug. It has been studied in adult T-cell leukemia lymphoma. And now we have some evidence of efficacy in relapsed refractory peripheral T-cell lymphoma in a prospective trial setting. T-cell lymphomas can be hard to study. They're more rare, it's harder to accrue numbers large enough for robust prospective randomized clinical trials. Sometimes for treatment, we're relying on case reports, case series, and retrospective data, which, although useful, can be harder to interpret and apply broadly. The Valentine PTCL01 trial is a multi-center open-label phase 2 study that enrolled 133 patients with relapsed refractory peripheral T-cell lymphoma, as well as 22 patients with relapsed refractory adult T-cell leukemia lymphoma. Very interesting. So 52 of the 119 patients with peripheral T-cell lymphoma that were evaluable had a confirmed objective response, and 17 of them actually had a complete response. It's about a 14% CR rate. 35 patients, or 29%, had a partial response, and the median duration of response was about 12 months. Median progression-free survival, 5.5 months. And then there was a post-hoc analysis. The median progression-free survival was out to 16.6 months for patients who achieved a complete response, and then 11.3 months for patients who had a partial response. And the estimated median overall survival after treatment with valametostat for peripheral T-cell lymphoma was 17 months. And these numbers might not look very exciting, but T-cell lymphomas are harder to treat than B-cell lymphomas, and their rare nature makes them hard to study, as I mentioned. So even with relatively modest results, this could prove to be a useful approach for some patients with relapsed refractory peripheral T-cell lymphoma, always looking for more tools to have in that particular toolbox. In terms of safety, of course, there were side effects, and common side effects included low blood cell counts, unsurprisingly, diarrhea, some patients experienced taste change and low appetite, nausea, and fever. The most common higher grade adverse effect was thrombocytopenia, and there were 13 patients of not a very high number who did stop treatment because of adverse effect. So just a word about safety of drug. At a first glance and digging into it a little bit, the patients did not experience a large number of what I would call dangerous side effects, but a number of patients didn't feel great on this drug. So an important consideration for any emerging therapy is whether we are extending progression-free and overall survival, but also whether that survival is with good quality of life. I don't want to pick on valametostat. It can be a good and useful drug. It's just always a good idea to have in our minds when considering any next line of therapy is, of course, are we buying time, but is it good time? Okay. Tazemetostat, also another one on the epigenetic front. This is in the same family of medications as valametostat, so it's a histone methyltransferase inhibitor. But tazemetostat is a selective oral inhibitor of mutant and wild type EZH2, and it's studied for treatment of non-Hodgkin lymphomas. There was a phase 2 study published back in 2020, and it showed activity of tazemetostat alone in relapsed refractory follicular lymphoma, so monotherapy. In that phase 2 study, there were 99 patients, 45 had mutant EZH2, and 54 had wild type EZH2. So one of the questions that study was looking to answer was, will this work in both mutated and wild type EZH2? Is it going to be better in one or the other? How did the patients do? And the objective response rate was 69% in mutant EZH2 and 35% in the wild type cohort. There were complete responses in 6 patients in the mutated cohort and 2 patients in the wild type cohort. So activity in both. Median duration of response was similar, 10.9 months in the mutant cohort, and then 13 months in the wild type cohort. And median progression-free survival was 13.8 months for the cohort as a whole. Reasonably well tolerated. Most common adverse events were nausea, diarrhea, alopecia, or hair loss. The higher grade adverse events were low blood cells, so neutropenia and anemia. And again, 8 patients did stop their treatment for an adverse event. So as is often the case, the drug showed promising activity as monotherapy. So it's being studied in combination to see if there is synergy. So whether all of the drugs combined work better than on their own. And tazemetostat is currently studied in combination with rituximab and lenalidomide for the treatment of relapsed and refractory follicular lymphoma on the SYMPHONY-1 trial. So phase 1B of SYMPHONY-1 showed that the overall response rate in the intention to treat population was 90.9% or 91%. So 40 of 44 patients responded. And in patients with wild type EZH2, response rate was 88.9%, so still very strong. In mutant, it was 100%, although I will say there were only 7 patients with mutated EZH2 and 36 patients with wild type, so a bit skewed there, but very interesting. The CR rate was 55%, and partial responses seen in 40% of patients, so very encouraging data, and the study is now in phase 3. So phase 3 is going to compare the intervention, so tazemetostat with lenalidomide and rituximab in combination versus lenalidomide and rituximab alone, which is already an established and frequently used regimen for relapsed refractory follicular lymphoma that's very effective. So a very interesting study, now in phase 2 and recruiting at various centers. All right. We're going to shift gears again and talk about bispecific T-cell engagers, likely a group of therapeutics that you're already familiar with or that you have heard something about. But just briefly, bispecific T-cell engagers, or BiTEs, or sometimes just called bispecifics, they've emerged as important treatments in B-cell non-Hodgkin lymphomas. They form part of our standard of care treatment for relapsed refractory diffuse large B-cell lymphoma. Examples of bispecifics that are used in lymphoma, drugs you may have already heard of, include glofitamab and epcoritamab. You may already know a bispecific T-cell engager has 2 arms, one that binds the cancer cell and one that binds cytotoxic T cells. Its job is to bring them together to efficiently kill tumor cells. This process of using those cytotoxic T cells and bringing it together with the cancer cell often results in an inflammatory cascade that we refer to as cytokine release syndrome, or CRS, and sometimes neurotoxicity, which we call immune effector cell associated neurotoxicity syndrome, or ICANS. All important things that we think about when starting treatment with a bispecific. So, new drug, AZD0486, which is now named, and it's called zorovatamig. It's an IgG4 fully humanized CD19 CD3 bispecific T-cell engager, and it binds CD3 with low affinity. So that is aiming to prevent an overactive immune response and reduce side effects like CRS. The hope is that we will still preserve the effective T-cell cytotoxicity against the malignant B cells. So there's a large study ongoing with many different sub-studies looking at a variety of CD19 positive B cell malignancies. To give an example of the efficacy, we'll look at the phase 1 study of surovatamig in relapsed refractory diffuse large B-cell lymphoma. So among the 40 patients who were evaluable, who received drug at doses of greater than 2.4 milligrams, part of early trials is figuring out what dose we need to treat people with in order for it to be effective but not overly toxic. So for those patients, the objective response rate was 43%. The complete response rate was 33%. And at what became the target dose of 7.2 milligrams, the overall response rate was 47%, with a 42% complete response rate. Median time on study of 7.5 months, still early for patients who received the target dose of 7.2 milligrams. Good. There was an estimated 12-month duration of response, so about 75% experienced a 12-month duration of response, and 12-month overall survival rate was 77%. Still very early, phase 1, small numbers, but interesting. They determined that the response is dose-dependent, and CRS rates did look to be low and low grade. So early signals that the low affinity binding to CD3 is helpful in reducing CRS. So far looking effective, but lots and lots of information to come on surovatamig. Okay. So we're going to change tack a little bit and talk about new combinations of established treatment. That doesn't mean that's the end of the emerging therapeutics. Certainly there are more, but in the interest of time, we're going to shift to see some new combinations of treatments that you may already be familiar with. So same as we've seen with new drugs. So tazemetostat was a good example. We talked about how there was evidence of efficacy in monotherapy, and now it's being studied in combination with lenalidomide and rituximab. So as we establish efficacy and tolerability for a medication, and it finds its place in sequence for lymphoma treatment, those that work well on their own often get rolled into combinations or moved earlier in line of therapy. We've seen with CAR T-cell therapy in diffuse large B-cell lymphoma. For refractory patients, we've moved it from third to second line, and there are upfront studies to watch out for. So now that we've established efficacy and safety of bispecifics, we're seeing them studied in combinations in a variety of disease sites and settings. So we'll talk about a few of those. So the first one to mention is glofitamab in combination with rituximab, gemcitabine, and oxaliplatin. You may have heard of the STARGLO study or have known patients who were a part of it. This was a phase 3 randomized open label trial that looked at the combination of glofitamab, so bispecific T-cell engager, and R-GemOx, a salvage regimen approved for relapsed refractory diffuse large B-cell lymphoma. And they looked at it in transplant ineligible, relapsed and refractory diffuse large B-cell lymphoma patients. Patients had to have one or more prior treatments. And after a median follow-up of 24.7 months, GLOFIT GemOx demonstrated a clinically meaningful overall survival, progression-free survival, and complete remission benefit versus R-GemOx, irrespective of the number of lines of therapy. So they did divide some of the data out by who had fewer and who had more lines of treatment, so who had multiple types of therapy before coming onto the trial. And it was, the addition of glofitamab to GemOx was better than R-GemOx on its own in all of those patients, though there was an improvement in patients who were receiving their second line therapy compared to those who were at third line or later. So patients were allowed on this study even if they've only had one treatment before. So you could come in on the heels of R-CHOP, for example. And for patients who were getting this treatment as their second therapy, the median overall survival was not reached compared with those in the R-GemOx arm. The median overall survival was 15.7 months, so a big difference there. For those patients who were getting this as their third line of treatment or later, so they'd had 2 previous therapies and now they were on trial or more, their median overall survival was 8.3 months. compared with 6.7 months if they got R-GemOx. So again, a really significant difference there. Median PFS also longer at second line. So second line patients, their median progression-free survival was 25 months versus 5 months for R-GemOx alone. And then in third line or later patients, progression-free survival, median progression-free survival, 9.2 months compared with 2 months R-GemOx alone. So better at all of the points and really quite impressive data. It was much better than R-GemOx alone. I would say R-GemOx is not a salvage regimen that we use frequently in Canada. We can use it, we do use it, but maybe have a slight preference for salvage with regimens you might have heard of, R-GDP and R-ICE, both with excellent efficacy data, but certainly an approved salvage regimen, and the evidence would suggest that R-GemOx is well tolerated. So patients who are not considered good candidates for autologous stem cell transplant, that's the other thing that this trial required, was that patients be deemed ineligible for an autologous stem cell transplant. But they may still be good candidates for CAR T cell therapy. So really important to assess whether CAR T therapy would be an option prior to choosing a regimen like this, but so good to have data for more effective regimens in the salvage space, where certainly we need to have options for patients. So there was cytokine release syndrome observed. It happened in 76 patients, or 44% of patients. Predominantly low grade. There were only 2 patients who experienced a grade 3 CRS, so a higher grade. Grade 3 CRS is typically treated in the ICU. If you've ever had experience with CRS or have a friend or a family member who has had experience with CRS, and grade 1 and grade 2 often can be dealt with on the floor. There were deaths related to glofitamab or rituximab. So they occurred in 5 patients in the GloFit-GemOx group and in 1 patient in the R-GemOx group. And I realize here I've labeled it GloFit plus R-GemOx, but when they use the glofitamab, they hold the rituximab. So it's glofitamab GemOx compared to R-GemOx. My apologies for the confusion. Also important to point out, other than the CRS, the common adverse events that I've listed here, certainly low blood cell counts, which we expect to see, so a reasonable amount of nausea and peripheral neuropathy, as well as diarrhea, and events for elevated liver enzymes. And there were some patients who needed to have dose adjustments as a result, but were able to continue on their treatment. Some patients did have to stop. As you might expect, because we're looking at something where we have a drug added to another regimen, the intervention arm had more adverse effect. Right? We're adding a powerful drug, a bispecific, to chemotherapy. So there were more serious infections in the GloFit-GemOx arm. There were 48 patients who stopped treatment in the intervention arm because of adverse event versus only 11 patients in R-GemOx. So there is a bit of a trade-off. There is definitely improved efficacy, but there is also some improved toxicity, so things to consider when we're thinking about regimens for our patients, for our friends, for our family. Okay, epcoritamab, and I will just put in a tiny plug, epcoritamab has also been studied with GemOx in a phase 2 study. So phase 2, epcoritamab, GemOx, no comparator, looking at safety and efficacy. We're not going to review it here because we've looked at glofitamab, GemOx. They're not the same drug, but they have some similar activity, and indeed it did seem to be an active regimen. What we're going to look at is epcoritamab and lenalidomide. So the rationale for the combination of these 2 agents is that given the converging effects on T cell activation and tumor suppression, the hypothesis is that the combination of epcoritamab and lenalidomide may further enhance tumor killing. So it showed promising activity in the initial phase 1b, phase 2 EPCORE NHL5. This is one of those large trials with lots of little sub-studies. The overall response rate for these patients who are relapsed refractory diffuse large B-cell lymphoma patients treated with epcoritamab and lenalidomide in combination. So overall response rate, 75%, complete response rate, 58.3%. And it seemed to have a manageable safety profile. So this is the early data. Now it's going to phase 3, and they are using R-GemOx as the comparator. So popular comparator arm for relapsed refractory diffuse large B-cell lymphoma. And it does get more use outside of Canada. It's a popular salvage regimen in the US and in Europe. So epcoritamab plus lenalidomide. Lenalidomide is a mouthful. Compared to R-GemOx, relapsed refractory DLBCL, stay tuned. For safety signals, the most common adverse events were low blood cell count, so neutropenia, anemia, low platelets. There were 2 patients who experienced febrile neutropenia, and many patients had CRS, predominantly low grade, but 10% of patients did experience a grade 3 CRS. So it was seen and important to consider. So very interesting. We'll be keeping an eye out to see how the phase 3 study goes. And as always, have an eye out for studies occurring near you. One more to chat about, just really briefly, so that we have time to have a bit of discussion. But I thought this was an interesting combination to talk about. So glofitamab, our bispecific antibody, with polatuzumab. Polatuzumab vedotin is an anti-CD79B antibody drug conjugate. Glofitamab, we've talked about, that's our CD20, CD3 BiTE. These drugs both have established activity in relapsed refractory diffuse large B-cell lymphoma. We use them already. Glofitamab is indicated in relapsed refractory diffuse large B-cell lymphoma. Right now we have access to it for patients who have received 2 prior lines of therapy. Polatuzumab indicated in new diffuse large B-cell lymphoma with RCHP. So there is a study supporting this. So it's not widely funded outside of Quebec yet. We will keep an eye and see what may come in future. But it's also indicated in relapsed refractory diffuse large B-cell lymphoma in combination with bendamustine and rituximab. And we use it in that setting for our patients who are deemed to be not eligible for an autologous stem cell transplant, and it's quite effective in that setting. So the combination of glofitamab and polatuzumab, the rationale is that glofitamab as a CD20, CD3 bispecific antibody engages and redirects T cells to eliminate B cells. Polatuzumab, an antibody drug conjugate which targets CD79B, has a complementary mechanism of action. So that could result in synergistic activity when combined. Good rationale. So there was further follow-up. So an initial open-label, multi-center phase 1b, phase 2 study was presented at ASH last year, and then more update has become available. So at data cutoff, they had 129 patients who had received more than 1 dose of the study treatment. And this is particularly interesting because this study includes diffuse large B-cell lymphoma and also high-grade large B-cell lymphoma. So not all of the studies that we look at for relapsed refractory large B-cell lymphoma includes those patients with high-grade B-cell lymphoma. So our high-grade B-cell lymphomas are the ones that have rearrangements of MYC, rearrangements of BCL2. These are formally referred to as our double and triple hit lymphomas, they're often, sometimes they're present in small numbers, but often not included in studies. So this had a good chunk of high-grade B-cell lymphoma, so very interesting. So there were 57 diffuse large B-cell lymphoma cases included and 44 high-grade B-cell lymphomas. There was also a little smattering of primary mediastinal and transformed follicular lymphoma, but we're going to focus on DLBCL and high-grade B-cell. 71% of patients included in this trial were refractory to their last therapy. These are heavily pretreated patients, tricky to treat. 22% had had prior CAR T-cell therapy. 50% did end up stopping their treatment, and 33% of those was because of progression, but some patients ran into trouble with adverse events, and we'll talk about that. But the best overall response rate was 80%, CR rate of 62%, and that was similar whether they had diffuse large B-cell lymphoma, sort of NOS, not otherwise specified, or if they had a high-grade B-cell lymphoma histology. So nice activity in high-grade lymphoma. In terms of follow-up, so they followed these patients. Right now, median follow-up is at 23.5 months. At the most recent update that I had access to, median progression-free survival was 12.3 months, median overall survival 39.2 months. And common adverse events include CRS. We are using a bispecific, which was mostly low grade, but there was one patient who experienced a death from CRS. It did occur in the context of an unresolved infection, and the patient had grade 3 CRS and then declined further CRS management, but clearly they were very sick. It does sound related to their treatment. 7 patients in the study stopped for adverse events felt to be related to glofitamab, and 7 stopped for adverse events felt to be related to polatuzumab. But very interesting data thus far, and a really interesting combination that in a tricky to treat group. So exciting to see what happens with that. So in summary, there are lots of exciting emerging therapies. There are new ways to combine established therapies in attempts to improve outcomes. Research is very exciting. Times like this, hopefully talks like this help to encourage people to look for ways to get involved. You're already coming to meetings like this, so clearly interested. Look for opportunities to be patient and family voices and advocates. It's so important. And ask your doctor, of course, about available and appropriate clinical trials. Thank you so much for your time this afternoon. I really appreciate it. And hopefully we have a few minutes for questions. Thank you very much.
Thank you, Dr. Murphy, for that overview in regards to therapies in the pipeline. As a reminder to the audience, at this time, you may submit questions in the chat box, which is in the middle of the black toolbar at the bottom of the screen. We'll try and answer as many as we can. So we did have a question come in. So after a phase 2-3 trial, how long can it take for the therapy to make its way into the market where it is finally available for patients?
Yeah, that's a really good question, and it varies. It really depends. It depends, but I would say it can take a year or two to finally make its way out. It's a little bit slower. Our regulatory processes in Canada are very thorough, and we're thankful for them, but they are a little bit slower. So I would say closer to the 2-year mark.
Okay. And you mentioned in the presentation that T-cell lymphomas are harder to study. Why is that? Is it just because they're a more rare subtype, or is there some other reason as well?
Yeah, that's exactly right, and it's a great question. But it is literally because there are fewer people. And one of the things that studies depend on is the power to be able to have results that are generalizable to a population. And it's really hard to power a study if you don't have a lot of people. And so it is easier to study T-cell lymphoma when you have lots and lots of centers involved. I'm going to say easier, not easy, because it's still a rare cancer. But the ways that we can do it is have big centers start studies. So big populated cities participate, and then lots and lots of participating centers from around the world. So global studies can be useful ways to study the rarer cancers, but it is because they are more rare.
Okay. And when is overall survival or progression-free survival counted from? Is it from the start of treatment or the end of treatment?
It will usually say in the studies. You can often find that in the bowels of the paper when you're looking through them, but often it's from the start of treatment. Sometimes it's from diagnosis, so you have to check and see how your statistician is calculating it. But try start of treatment or time of diagnosis.
And given the rapid pace of research, do you foresee a point where cellular therapies or targeted therapies could become a frontline option for more patients?
Yeah, what a great question. And I do think it's possible. Cellular therapies in general are being studied upfront in a variety of heme malignancies. And certainly we've seen how important they are to treatment. We're fortunate to be able to study them. And I do think that they will make the effective therapies tend to move earlier in line of treatment. So with some good studies showing that they're better than what we already have, well, that's how they'll move to frontline, right? If they can show that they beat what our first line treatment is now, then that's the way that we will go.
Okay. And going back to the first question on phase 1, phase 2 trials, we do have a session on clinical trials at the very end of the conference session today. So I advise you attend that session for more information. And I do want to now just direct the audience to attend the next breakout room session according to your subtype. So there's a few sessions happening at the same time, including Hodgkin's lymphoma, diffuse large B-cell lymphoma, CLL, follicular lymphoma, mantle cell, Waldenstrom's, and cutaneous T-cell lymphoma. So please make your way according to your subtype. We do have a 10-minute break before the next session begins. And I know it can be difficult on the body to stay seated for a while, so I recommend perhaps you take a standing break, and then after 10 minutes, we'll see you at the subtype sessions. Thank you, Dr. Murphy. Thank you everyone for joining, and we'll see you in the next session.
Thank you so much.